Hi everyone, we're out on my front porch again, and tonight I'm going to take you through dissecting the sheep brain. And so wherever you decide to dissect, I've picked a plastic folding table that we keep out on the porch. You want to start off by cleaning it with a multi-purpose cleaner or some dish soap, okay? Start off fresh and clean, okay? And from here, you're going to want to throw some gloves on. I have a bag for trash. Just as I go, I'm going to throw things in there. The brain itself, I'll put in a Ziploc bag when I'm done with it, and then put it in this bag, tie it up, and put it in the trash. The brain is shelf stable and it's very safe. It has far less than 3% formaldehyde in it. It's usually under 1%. Um, and so, you don't want to eat it, but it's all right. So I have a dissecting tray like you would see in a lab, and I'm just going to cancel out of the thing that just showed up on my screen. All right, so we have this nice tray of dissecting tools. I'm actually going to move the computer so that you can see my tray a little bit better, hopefully. Okay, off the duck. All right, so we have the tray, and we have brains. Um, so I have a couple brains in here. So I'm going to take this one out. Just going to make sure that you are good. Okay. So first, before I go into the brain, I want to go through my tools. So I have a scalpel like you do. I have dissecting scissors, which did not come with your kit, but if you have small scissors that you don't mind washing, you're welcome to use them. I have a maul probe, which is just a fancy metal pointer. It can also be used to kind of separate tissues. And I have forceps, and forceps are good for holding on to things or picking little parts out. So we're going to look at the sheep brain a little bit. So here, this covering is the dura mater. Okay, and some of you are going to have dura mater that has a little bit of skull left. It is what it is. And so the dura mater means tough mother, and so it's one of the meninges. There's the tough mother, the dura mater, the arachnoid mater, which is the spidery mother, and it looks kind of like web, and then the pia mater is the soft mother. Okay, on the inferior side, we have some fun things to look at. So at the anterior inferior side, let me get this close, you probably have two openings, and those openings are going to show you what are called the olfactory bulbs. So these bulbs are actually found in the back of your nose. And from the olfactory bulbs, I'm just going to cut a little bit so you can see them. You will see coming down olfactory tracts. So here is the, let's see, here's the olfactory bulb, and down there is the olfactory tract. That's cranial nerve 1, okay? And we can also see cranial nerve 2. And so here's part of it, and here's part of it. It usually has some fat around it. And these nerves go to the eyes. These are the optic nerves. And then the optic nerves are going to cross at the optic chiasma. So the optic chiasma is right here where the optic nerves cross. Okay, hopefully you guys can see that. Right there. And so your right eye crosses over to the left hemisphere, your left eye crosses over to the right hemisphere. And below that, you have this little nose, and this little nose is the pituitary gland. Okay, so it's the pituitary gland. Some of you may be able to see some other nerves. I can see, so it's TTA. I can see the abducens nerves. And so that's the way I remember it. So cranial nerve 1, 2, and 3 start with T, T, A, and then it's oo, 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 and then you go from there. And this over here is part of the trigeminal nerve, which is this cool nerve that kind of comes out, goes over your mouth, over your eye, and under your mouth. So I will expect you to know the the olfactory bulb and the optic nerve and the optic chiasma. All the other nerves on the sheep brain, they're really inconsistent about what you can see, so I'm not going to expect you to know any of those. You should definitely know this pituitary gland because it's so important. And so before we do any cutting, I want you to take a picture of the superior and inferior parts of your brain so you can label it. So we can see the right and left hemispheres here. I should say right and left. And behind that, we have, this is the cerebellum. So this is the cerebrum and this is the cerebellum. And so I'm going to start by cutting just to the side of the longitudinal fissure, cutting off the dura mater. So you just have a scalpel. So I'm going to show you how I do it with a scalpel, and then I will show you how to do it with scissors. Okay, so you just take the point of the scalpel and you have to cut in. And so you want to, this is like a seam right here, so you want to stay close to the seam so you're not cutting into your brain. Okay, as much as possible. Some of you will cut into your brain. Try not to go too deep, okay, because the deeper you go, the more likely you are to cut into your brain. And so once you've cut through, you can kind of look at what you have. So now you can see, oops, I cut through some of the brain. It's okay, the sheep's dead, he doesn't care. All right, so just kind of keep cutting. So here, I actually like to use the scalpel with the blade up. And when you're using it with the blade up, never have your hand over it, never have your face over it, and always point it away from you. But I like to kind of get the tip and kind of tear the dura mater as you go. But now I'm getting close to my hand, so I'm going to stop doing this. Okay, and you can just kind of pull some of it off. I'm going to take the scissors because I have some scissors, and I can pull some more off. Now, if you just pull the dura mater off really quickly, you're going to lose all of these nerves, and you're going to lose the pituitary gland. You may lose them anyway, but the more careful you are with these, the better. Okay, so I've gotten the brain, the cerebrum, out of the dura mater, and now we need to cut. We need to cut the tissue that's between the cerebrum and the cerebellum. Okay, and so you can take the scalpel, and I'm actually going to go on the inferior surface, and I'm just going to use that tip and try to cut the dura mater off. So here's the pons and the medulla oblongata. I'm trying to cut the dura mater off right to the side. So I'm hoping to leave this stuff intact, but get the dura mater off the rest of the brain. Okay, and just go slow and steady, and make sure you never put your finger under the scalpel. Okay. Right, so I'm making some headway here. You know, it is really accurately named the Tough Mother. Okay, and I'm just going to cut the rest of this very quickly, just so this video doesn't take longer than your attention spans. All right, so now I'm removing the dura mater, and you can see the cerebrum and the cerebellum clearly. Okay, got a good chunk there. You'll see that scissors really are much better. I just didn't want you guys to have to buy scissors. All right, so we have the cerebrum and the cerebellum, and you know, we can still see the parts over here. I was able to maintain those. We have the pons up here, the medulla oblongata going down to the spinal cord. And I'm going to, if you follow the instructions that I posted for you, it says to kind of bend the cerebrum. And if you do this, you have to be careful because the cerebrum can just break off. Okay, so I'm going to carefully do this, and I'm actually going, I need to see a little bit. So if you do it just enough, it's going to look like a little nose is sticking out between the cerebral hemispheres. And I am having moderate success here. You can kind of see it right there. Okay, that's the pineal gland. That's the gland that produces melatonin and controls our day and night cycles. Below the pineal gland, you'll see two big bumps right here and two little bumps. Those are it's the bodies of the four twins. Corpora means bodies, quadra means four, twins is gemina. Corpora, quadra, gemina. Okay, so far we have the right and left hemispheres, longitudinal fissure, the cerebellum, the pineal gland, and the corpora quadrigemina on this aspect. All right, so now it's time to cut the brain in half along the longitudinal fissure. Okay, I'm just going to make sure that everything's still recording. All right, and so I'm going to come down, and actually I always start on the anterior part first. I'm going to come, and so you don't want to, you're not Jack the Ripper, right? You're going to take long, smooth strokes. Okay, so we're going to, and deep, but not too deep. If it feels like you're really tearing it apart, just take it easy, right? Lay off. Okay, we're going to do this one more time. This one just came off so nicely. I'm going to see if I can get the whole thing in half because I have so many structures on the bottom. And sometimes it takes more finesse than that, but this time it didn't. So, yay for me. Okay. And now I'm going to cut the cerebellum. Okay, and the cerebellum can sometimes be a little mushy. Yep, I just had a big chunk of cerebellum come off. All right. So, what you see now should look something like this. And I'm going to go through all these structures that you see here. And let me just compare my two sides. Sometimes one side is a lot better than the other. Well, they're more or less the same. All right. So, let me get up close. Here, I'm going to move this stuff out of the way. All right. So what we can see here is that we can see up here is the cerebral cortex. That's the gray matter on the outside. We can see these bumps. The bumps are called gyri. The dips are called sulci. And so, just kind of get a feel for these. The reason why we have these in our brain is because it increases surface area. So you can pack a lot more cerebral cortex in a smaller space. Folds almost always are there because they increase surface area. So the gray matter is mostly cell bodies. Okay, white matter is mostly axons. And so when we cut through this, you're going to see white matter in this part of the brain. But right now, you see white matter right here. Okay, and that's a special white matter called the corpus callosum. And it is what allows the right and left hemispheres to talk to each other. Below the corpus callosum is another piece of white matter called the fornix. And in between the corpus callosum and the fornix is the area called the lateral ventricle. This is where we have a right and left lateral ventricle, and this is where cerebral spinal fluid flows. Okay, those ventricles are separated by tissue that's called the septum pellucidum, and I have just a little bit of septum pellucidum right here. If you are somebody who likes to kind of dive in and you go deeper in, sometimes you'll be able to see some red capillary beds, and I actually can't see it in this one. Sometimes you'll be able to see some like reddish brownish capillary beds, and those are called the choroid plexus. The choroid plexus is what makes cerebral spinal fluid. It's capillaries that force out plasma that turns into cerebral spinal fluid. Okay, so we have the corpus callosum up top, we have the fornix on the bottom, and we have the lateral ventricle right here. Okay, the next things that we're going to look at are the epithalamus, the thalamus, and the hypothalamus. So let's start at the thalamus, and the thalamus is this egg-shaped structure right here. Okay, and some of you are going to have a spot inside the egg-shaped structure, can't speak anymore, and that spot is called the interthalamic adhesion. Okay, and we're going to talk a lot more about the thalamus and the hypothalamus as things go on. Okay, so in front of the thalamus, so this is the thalamus, and in front is this area is called the hypothalamus. And the walls of the hypothalamus forms the walls of yet another ventricle. So just kind of keep that in the back of your mind. So here's the hypothalamus, here's the thalamus, and then up here where that pineal gland is the epithalamus. Okay, so we have epithalamus and hypothalamus. Okay. Other things that we can see here, we can see it looks almost like a tree inside the cerebellum, and that's the tree of life, the arbor vitae. Okay, and I guess we should hang out with the ventricles now. So we've talked about the lateral ventricle. It's very hard to get the pointer where you want it. So there's the lateral ventricle, and we talked about the hypothalamus forming the third ventricle. And so if you go back here, connecting the third ventricle to the fourth ventricle is the cerebral aqueduct. Okay, let's see if I can really, let me see if it's better on this. Ah, yes, it is better on this brain, or this hemisphere. Okay, so right here, that's the fourth ventricle. And the cerebral aqueduct is like this area right here that connects the third ventricle to the fourth ventricle. So cerebral spinal fluid is made in all the ventricles, in the choroid plexi of all the ventricles, and it travels from the lateral ventricles to the third ventricles, down the cerebral aqueduct to the fourth ventricles, down the center of the spinal cord, up and around, it goes around the brain. And it's, it has a circulatory pattern, but it's not like blood vessels where, there's a very specific pattern. This is just the general current that it goes through. Okay, so we've covered all of these parts of the brain. And I'm like Julia Child. I'm just going to take this beautifully baked lasagna out of the oven. No, I have a brain that I was playing with earlier today. And so I'm going to take that out because it has some interesting cuts to it. Let's see if I can put it back together. Best jigsaw puzzle. Okay, so I cut the brain. I did a frontal section, and so this is just the right hemisphere, and then I did a frontal section of the right hemisphere. And so I want you to be able to see, so this is that gray matter on the outside, that cerebral cortex, and then there's white matter on the inside, and that white matter again is axons. And the axons allow the hemispheres to speak to each other, allows the different parts of one hemisphere to speak to itself, and allows the hemispheres to speak to the spinal cord. Okay, so that's where the signals are being sent. You can also see a bit of the lateral ventricle. So the lateral ventricle actually is really deep. My daughter's old Daniel Tiger thing just went off. I don't know if you heard that. All right, so the lateral ventricle actually is pretty deep. Then it forms something called it's a horn, okay? And so from here we can see also the corpus callosum up here and the fornix down here. And then, so this is that frontal section. I also did a transverse section. Okay, and so now, we're only looking at a quarter of the cerebrum, but now you can see what it would look like if we did a frontal section of the brain. And over here, we're seeing some gray matter on the inside of the brain. And these are the nuclei. On the outside of the central nervous system, we have masses of cell bodies that are called ganglion. Remember that dorsal root ganglion? So here we have nuclei. Okay. So just to wrap up, I hope that this is helpful in helping you get through dissecting your sheep brain. You want superior and inferior photos before you cut. You want to remove the dura mater. Ideally, you'd get a photo of what happens when you pull that cerebrum down, and you can see the pineal gland and the corpora quadrigemina. Okay, so you label those. You'll cut the brain in half, and I want the thalamus, the hypothalamus, and the epithalamus labeled. The corpora quadrigemina, sorry, the corpus callosum, not the corpora quadrigemina. Corpus callosum, which again allows the right and left hemispheres to talk. The lateral ventricle, the fornix, the cerebral aqueduct going to the fourth ventricle. Okay? Over on the inferior side, you should have the olfactory bulbs and the olfactory tracts, the optic nerves and the optic chiasma, the pituitary gland, the pons, the medulla, and spinal cord. Okay? So that's just kind of going over the information that you already have, but I just wanted to make sure that you saw somebody do it. So I am now done. So I'm going to wash my tools because I will reuse these. These are reusable things. Your scalpel is a disposable scalpel, so you can just put the little cover that it came with on, and then you can dispose of it. I am wrapping up this brain in a Ziploc bag, and the brain is going into my target bag. Okay, and just as I said, I'm going to tie up the target bag and do away with it. I have some more cleaning to do, so I don't want to take off my gloves yet. But once I'm done, I'm going to take off my gloves and I'm going to clean the surface again with a multi-purpose cleaner or dish soap if that's what you have. Okay? I hope that this calms any of your nerves. I know that this is probably a little weird dissecting at home, but it's really important to get an understanding of what this structure really is like, that kind of how it feels, what it looks like, like that. It just gives you an understanding that looking at pictures or models doesn't give. Please post questions to the community help forum if you have any questions. And otherwise, I'll see you at my office hours or the next time you're online. Okay? Hope you guys have a good night. Have a good night. Bye-bye.